One of the more promising potential therapies in the war against the coronavirus is the use of plasma from people recovered from the virus. Convalescent plasma, the liquid part of blood, is rich in antibodies. Although the treatment is early in the experimental stage, early reports indicate that injecting those antibodies into people with COVID-19 might give their immune system a much-needed head start, boosting their ability to fight off the virus, preventing the illness from becoming more severe and perhaps hastening their recovery. This kind of therapy, called passive immunity, has been around for more than a century. Doctors employed it against the 1918 flu and against polio and measles before vaccines for those diseases were developed. Scores of convalescent plasma clinical trials are taking place in the US and around the world, comparing people with COVID-19 who receive the plasma to patients who don't. The U.S. Food and Drug Administration (FDA) has also approved an expanded access program in which people can receive plasma outside of studies if they've been admitted to an acute care facility and consent to the treatment. More than 19,000 people have received plasma under that program, and early data suggests that the treatment is safe. Whether it's effective against COVID-19 remains to be seen. More than any substance, plasma exemplifies the promise and perils of modern medicine. The tea-colored liquid was first separated from blood more than a century ago. Today it serves as raw material for an international pharmaceutical industry that's worth $20 billion and growing. Every year plasma companies in the U.S. collect more than 38 million pints for processing into more than 50 therapeutic agents, including antibodies, clotting factors, and other useful proteins. They also pay people to obtain it. Most other countries don't allow such arrangements, but it's always been the norm in the US. As such, we've become the biggest player of the plasma trade. Plasma is easily contaminated by bacteria, and if it's processed improperly it can kill someone just as easily as cure them. But there's a dark side to America's dominance of this resource. Although similar in some ways to donating blood, plasma collection is time-consuming and inconvenient. Giving plasma takes an hour and a half versus the 10 minutes for donating blood, and involves inserting a needle into each arm instead of just one. During the process, blood is drawn from the donor, the plasma is separated and the red cells re-injected. Because of the added time and discomfort, collectors generally pay donors for plasma donations, although not in the case of the COVID-19 plasma donations. Even with the incentive of cash, usually $30 to $50, most gainfully employed people are not willing to put up with the time and discomfort. Because of that, most collection takes place among people who need money, in low-income neighborhoods, a morally ambiguous situation at best. So while plasma-related therapies have saved countless lives, they've always been shadowed by a sense of exploitation and scandal, and human tragedy. The story of plasma begins on the eve of World War II, the greatest blood shedding in human history. The Allies knew that blood would be a key resource for treating those injured in battle, but were also well aware of its shortcomings. Blood could only be stored for a few weeks before the red cells break down, too short a time to be shipped and saved for battle overseas. They needed to find a component of blood that was more durable. The most obvious choice was plasma, the amber-colored mixture of water, protein, antibodies and clotting factors that rises to the top when whole blood is centrifuged. Plasma cannot serve all the functions of whole blood, lacking red cells, for example, it cannot carry oxygen. But in wartimes it could serve as a substitute when people needed their blood volume restored quickly, as in burns or crush injuries. And unlike whole blood, plasma could be stored for months and could handle the rough conditions of wartime air transport. That knowledge became crucial early in the war, when Americans began shipping plasma to London. With the German Air Force pummeling their city, the British were in desperate need of transfusions. Led by Charles Drew, 1500th, a prominent African-American doctor, the Red Cross, and other organizations mounted blood drives in New York and set up a system to separate the plasma and ship it abroad. It was no easy task. Plasma is easily contaminated by bacteria, and if it's processed improperly it can kill someone just as easily as cure them. For that reason, the British referred to plasma both as liquid dynamite and liquid gold. By the time the project ended in 1941, Drew and his colleagues had successfully collected, processed, and shipped 5,500 units of the life-saving liquid. Drew became a revered figure in American medicine. A gifted athlete and scholar, he was the first African American to earn an MD at Columbia University, and a revered educator at Howard University before taking charge of plasma for Britain and later returning to teaching. He's equally famous for the body of myth surrounding his death. Gravely injured in a car accident on the way to an annual free clinic in the Deep South, he was taken to hospital in North Carolina, where some say he died because the doctors there refused to give him blood from white donors. In truth, the doctors recognized Drew and did everything they could to save him. Other researchers found that freeze-drying plasma made it even more stable, giving it an indefinite shelf life. When American medics deployed overseas, they carried packets with a vial of dried plasma along with a vial of distilled water to reconstitute it for the wounded. The Germans had none of this technology. They killed or drove out many of their best scientists, who were Jewish, and focused their blood research on proving the racist myth of Aryan superiority. By war's end, plasma and blood had become critical parts of the American treatment arsenal. But that was only the beginning. In the post-war years, plasma became a key raw material for a burgeoning pharmaceutical industry. And as the industry grew, its leaders found it impossible to maintain their supply. In the 1960s, a new process changed the game. The process, called plasmapheresis, involved hooking the donor up to a machine that separated the plasma and returned the red cells to the person. People who donate whole blood can only do so 6 times a year, because it takes time to regenerate red cells. To donate more frequently would cause anemia. But plasma regenerates in a few days, meaning a plasma donor can safely donate 104 times annually. To the pharmaceutical industry, this was the equivalent of hitting a gusher, and the industry took off. Treatment with plasma became a standard practice in surgery. Rather than infuse whole blood into patients, surgeons could infuse plasma. Antibodies derived from plasma became an important source of passive immunity, the same concept that's being explored to fight the coronavirus today. The technology soon became a blessing to people with hemophilia, a genetic condition that prevents blood from clotting. For most of history, these people would die of hemorrhage before reaching adulthood. In the early 20th century, they would get whole blood transfusions, usually from family members, but with little effectiveness. Things improved in the 1960s, when patients could go to the hospital for intravenous infusions of plasma or its concentrate. But in the 1970s, a miracle arrived. Scientists developed a plasma-derived concentrated clotting factor, called Factor VIII, that patients could keep in their refrigerators and inject with a syringe whenever they needed, in case of an internal hemorrhage or external bleed. It changed hemophilia patients' lives utterly, doubling their average lifespans and giving them the active lifestyles they wished for. Corey Dubin, a radio producer and activist with hemophilia, who I interviewed in the late 1990s, told me that before Factor VIII concentrate came along, he always had to be within a short drive of his hospital, where he could rush to get infusions of plasma in case of a bleed. After the concentrate, he felt like a free man. He could hike the mountains of California and bushwhack through the jungles of Costa Rica, while packing some concentrate and a syringe. Just as the oil industry explored the world for places to drill, plasma collectors went prospecting for new supplies. But there was a hitch: each unit of Factor VIII required many units of raw material to start with. Even by bleeding donors more than 100 times a year, the plasma collectors could not get enough. And so, just as the oil industry explored the world for places to drill, plasma collectors went prospecting for new supplies. The obvious place was in America's poor neighborhoods, where many plasma collectors set up their centers. Many of those centers were unclean and poorly regulated. Some paid donors in chits redeemable at the local liquor store rather than in cash. In the mid-1970s, a series of newspaper and television reports revealed the degrading conditions in some collection centers. In response, many collectors moved to the developing nations, notably, Haiti and Central America. "We were peddling so much plasma out of Costa Rica that it must have been their second biggest in export after bananas," one pharmaceutical company executive told me. That didn't last. When newspaper reporters exposed the center in Haiti, the country's dictator ordered it destroyed. A few years later, the collection center in Nicaragua became such a hotbed of anger that rioters torched it, triggering that country's revolution. Worse scandals were to come, as some collectors moved their business to prisons. One collector, Austin Stuff, MD, had gained access to several prisons in the South through political connections and bribery. Stuff's target was high-priced immune globulins, the antibody-containing component of plasma. Antibodies are specific to disease. In order to get antibodies for measles, for example, you have to obtain plasma from a measles survivor. Rather than scour the country for survivors, it's more efficient to inject a potential donor with a low dose of the disease and then return to collect his plasma after it's become rich in antibodies. To Stuff, prisons made ideal harvesting grounds. The inmates were eager to make a little cash, usually very little, and it was easy to find the same donor on the return visit. His business thrived until the mid-1970s, until his sloppy practices and failure to sterilize equipment led to a series of hepatitis outbreaks that caused him to be shut down. But others succeeded him, and prison collections continued into the 1990s. By the mid-1980s, dealers like Stuff were fast disappearing, and collection centers tended to be well run and clean. Europeans scorned America's for-profit collections, while quietly and hypocritically importing most of their raw material from the United States. But even during that prosperous time, conditions were lining up for a tragedy. Two factors set the stage. One was the collection in poor neighborhoods, where people generally had higher rates of infectious disease, such as hepatitis. The second was the way that the plasma was handled. In order to efficiently process the material, the plasma was consolidated into enormous pools, sometimes of up to 60,000 donors. This caused a tremendous mixing to take place, especially with the more concentrated medicines, which required greater amounts of raw plasma as the starting material. Virally speaking, someone with hemophilia injecting factor VIII might be making contact with thousands of other people. With highly concentrated material being shipped internationally, a pandemic spread by plasma products was bound to occur, and it did with the spread of hepatitis and AIDS. Most of us know the story of AIDS, how it first appeared in the gay community, early on was ignored by politicians, and then spread throughout the world through sexual contact and blood transfusion. What's less widely known is how it devastated the hemophilia community. In what became known as the hemophilia Holocaust, thousands of people became infected because the medicine they took was contaminated with HIV. Dubin told me that he had long suspected he'd been injecting himself with contaminated factor VIII. I decided that maybe I could survive it, he said, when we spoke in the mid-1990s. He died of AIDS in 2017, at the age of 61. Thankfully, the plasma is safe now. In the wake of the AIDS epidemic, scientists developed ways to deactivate plasma-borne viruses and transition to using smaller pools. More recently, they have used genetic engineering to develop factor VIII and other products that do not rely on raw human plasma. Factor VIII has returned to being the blessing that it once was. Today, we find ourselves at the threshold of a new chapter in plasma-based therapy. Thousands of people are receiving convalescent plasma in the hope of fighting COVID-19. Many people with infections are taking part in clinical trials, calibrated experiments comparing patients who receive plasma to patients who don't. Other people with severe COVID-19 qualify for compassionate use, receiving the experimental therapy on an emergency basis. So far, the reports of how the plasma therapy is working are anecdotal, and we won't see definitive results for months. Early reports from the Expanded Access Program indicate that it's safe for patients suffering from COVID-19 to receive plasma from those who have recovered from the disease, although the effectiveness still needs to be calibrated. If successful, this treatment could hold the line until vaccines or cures have been developed. And once again, plasma could become widely known as liquid gold.